Hello everyone. Today I'm going to explain about DNA replication in prokaryotes. I'm doing this only for some students who are focusing only on board examination and JEE students, because no need to learn each and everything related to replication. So it will be a burden for them. So based on your request, I'm going to do it again. But please pardon me that I'm using pen and paper method and using a old mobile phone where the clarity is not that great. Hope you can see it. So we'll start with the DNA replication in the prokaryote. The first thing is I'm going to write important points here, the requirements. First is the oocyte. In the case of E. coli or the bacteria, that is prokaryote, there will be only one oocyte. There will be only one oocyte. Okay, so in the case of eukaryotes, there will be many, that is multiple oocytes will be there. But I'm not going to talk about eukaryotic replication. I'll stick to only this one, E. coli, that is bacteria. Prokaryotic replication, oocyte means origin of replication. So if I draw a DNA of prokaryote, so these are all the nitrogenous bases, correct? Okay, which joins the double strand, two strands, make it double stranded structure. And here what you could see is there is somewhere else a point where some sequences of nitrogenous bases where the DNA enzymes that is required for replication process will bind. So this point where in the DNA is called as the oocyte, and this is the point where replication initiates. Okay, so this is ori means origin of replication. Okay, now another important enzyme required for this replication is helicase, and third important enzyme is the gyrase, and the fourth important protein is single stranded binding proteins, or in short we can also write this as SSBP. Single stranded binding proteins are required for it. So the next one is we need RNA primase enzyme. The main function of them is they will bring the ribonucleotides, that is RNA nucleotides, and we call it as primers or RNA primers, both are correct. And next what is required is very important enzyme for the polymerization of the DNA strand. That is, they will add the deoxyribonucleotides and make a long chain of the new DNA strand. That is called as DNA dependent DNA polymerase. I will write only DNA polymerase. Okay, so DNA polymerase main function during replication is it will bring DNTP. What is this DNTP? D means deoxy, that means something related to DNA, deoxy ribonucleic, correct? So deoxy nucleotriphosphate. Okay, so we just write abbreviation of it, deoxy ribo. Okay, I forgot ribo to mention ribo. Deoxyribonucleotrophosphates. Okay, so that will be brought by this enzyme called as the DNA polymerase. Chalo, it's very simple. Then finally we need one enzyme that will join the separated deoxyribonucleotides. It is called as the DNA ligase. So please remember these 7 important points. You need to mention all in the examination. So that's what, like, okay, so we'll start with the oocyte now. This oocyte will attract helicase enzyme. Okay, helicase enzyme will bind somewhere else very close to the oocyte. Okay, so helicase enzyme will bind. The main function of the helicase enzyme is to separates or unwinds DNA strands. Now DNA strands are like, it's joined by nitrogenous bases, right? How to separate them? Very simple. I need to break these nitrogenous bases, correct? Simple. So if I break the nitrogenous bases, how can I break the nitrogenous bases? By breaking or removing hydrogen bonds, correct? So we know that nitrogenous bases are linked by hydrogen bonds. So I can remove the hydrogen bonds by adding helicase. So helicase will now remove the hydrogen bonds. What is the function? It removes hydrogen bonds Got it? Okay, so when hydrogen bonds are removed, what's going to happen now? So we'll draw the diagram now. Okay, so this is now the DNA. After the action of the helicase, you get a replication fork. A fork like DNA will be seen, and this structure is called as replication fork, formation of replication fork. So that what you could see is separated nitrogenous bases, but here the helicase action is not seen. Still this DNA is now double stranded, but in this case actually they are separated because of helicase enzyme. Okay, so this is very important, removes the hydrogen bonds between the nitrogenous bases, and now another function is it separates the DNA strands. Done. So it's very easy, right? It's very easy, right? Okay. After the action of the helicase, when the DNA strands are now being removed like this, pulled to the other end, so the pressure will be building on this part of the DNA, that is the upper portion of the DNA. So this leads to supercoiling. So what is this supercoiling? Supercoiling means already DNA is helically coiled. We know it. DNA will be like this, helically coiled. So when I start pulling this to this end or to this to the other end, what's going to happen? The pressure will be built on this, correct? So that see this. See the distance between the nitrogenous bases now reduced because of the pressure. This pressure is called as torsion. So torsion pressure. Okay, torsion will leads to supercoiling of the DNA. Okay, so we need somebody to prevent this supercoiling or torsion of the DNA on the upper portion of the DNA. So that is the second enzyme, that is gyrase. So what is the function of it? It prevents torsion or prevents supercoiling. So you need to write this. Next is, once the DNA strands are separated by the helicase enzyme, so here another type of protein called as SSBPs, that is single strand binding protein, will bind to each strand. See here, SSBPs now bind to each strand, that is separated strand of the DNA, and which now prevents joining of separated strands. I think I'm writing fast. Okay, prevents the joining of separated strands. Remember this point. Okay, so this is the important thing. Okay, how they prevent? They will not allow the formation, that is reformation of hydrogen bonds between the separated nitrogenous bases. One thing. And other thing is, second important function of the same SSBP, this is first function. Second is, it gives stability to the separated DNA strand. See, this is now separated DNA strand. Okay, so how it provides stability? Because when earlier the DNA was like this, it was like helix. Okay, so once you separate it, it tends to coil around itself. The separated strands tend to coil around itself, like it looks like this. So we do not want this structure. We want a structure like this, very long and narrow structure, linear. This is actually coiled, kind of like semi-coiled, because it's the habit of the DNA. Even though you separate the strands of the DNA, they tend to coil around itself. Okay, so that is actually prevented by the same proteins called as single strand binding proteins. So that is the function, second function of it. Okay, now another thing is RNA primase. Okay, so this RNA primers or primase enzyme brings, so we just write its polarity. Here I'm going to write 5' end, that is the starting strand. This is the opposite end of it, so 3'. What you see here? You see a free phosphate group, we know it, and here you see a free OH group. In this case, it is the anti-parallel strand, that is opposite strand. So it should be having opposite polarity, 3' to 5'. Correct. So very simple now. So here, exactly here, very close to the 3' end of the DNA strand, RNA primers will come and they bring or add a type of ribonucleotides. What is this ribonucleotides? RNA primers will add ribonucleotides, that is, we'll say 1, 2, 3, 4. That is the nucleotides of the RNA. I did not write deoxyribonucleotides. If I write, then obviously it should have been deoxyribonucleotides. So, but it is brought by the RNA primase enzyme. So it should be RNA primers. The primers means the unit which contain adenine as nitrogenous bases, guanine, cytosine, uracil. Instead of thymine, I am writing uracil. So I am going to join it. So if I join it, now it is nothing but ribonucleotide chain of the RNA, correct? So now it is a ribonucleotide chain. Normally the ribonucleotide chain which is now brought to the 3' end, see 3' end of the DNA by RNA primase enzyme. Okay, so that contains around 22 nitrogenous bases. How many? Maximum 22. So it's a very small chain, so which will be brought here. Now this is the initiation of the replication. Okay, so this is now I'm right primers. What is primer now? Primer is nothing but ribonucleotides brought by RNA primase enzyme. Where? To the 3' end. Once it is done, another enzyme here, that is DNA polymerase. What is the function of DNA polymerase? It has two functions. The one function is it will bring DNTPs to the site where the primer is been added. That is here, somewhere else here. Okay, so now it's going to bring the DNTP. What is this DNTP? DNTP means, as I already mentioned, deoxyribonucleotriphosphate. Okay, DNTP. What is this DNTP? DNTP is a long chain of this DNA nucleotides that contains DNA nitrogenous bases like adenine, guanine, cytosine, thymine, adenine, thymine, like that. Got it? So a long chain will be brought and it's going to continue like that. This is actually the direction. So that means the replication occurs from 5' to 3'. That is, I'm talking about a new strand, correct? So this is the new strand, right? This is the new strand, right? So see the movement, once the primer is added, then DNTPs are added in this direction. So see, that is the reason why I said DNA replication occurs in 5' to 3' end. So please do not consider this 3' to 5'. Okay, so only you have to consider the new DNA strand, that is 5' to 3'. You may get a question, where exactly the primers are added? That is very close to the 3' end of the DNA. Got it? So this is the thing. Now DNTPs are added by this DNA polymerase. Okay, so many DNTPs will be added like this. Many DNTPs will be added, and finally a long chain of new strand of DNA is synthesized. This process is called as polymerization. So what is polymerization? It is the process of synthesis of new DNA strand is called as polymerization. What is the meaning of polymer? Polymer means a long chain. Correct, a long chain of the DNA, that is polymers. Even the protein also a polymer, correct. So it is made up of different types of amino acid. Done, very simple. Okay, once it is done, so we have to go to the other side. Here, due to, see, the reading frame is 3' to 5'. In this case 5' to 3'. So it is very easy for the primer to detect the 3' end. So they will start adding the primers. One primer will be added, only single primer will be added, and after that DNTPs will be added. I forgot to mention one more thing. What is the other function of DNTP? When DNTPs are brought by the DNA polymerase, it also provides energy in the form of ATP. Okay, done, very simple. So two functions of DNTP. One is it will initiate the polymerization of deoxyribonucleotides, and second function is it will also provide energy to the polymerization process for the DNA polymerase enzyme. That is the thing. In this case, when you read the strand, that is 3' to 5', it's very complex for the RNA primase enzyme to recognize the 3' end, because the 3' end is somewhere else here, correct? It's very far. So it has to add the primer to somewhere else here. Okay, so here it leads to confusion for the RNA primer. So because of this confusion, it will add many primers. See how many primers were added to this end? Only one, by the primase enzyme. But in this case, due to the complexity in the polarity of the strand 3' to 5', so it will add many primers like this. So these primers will be added. Once the primer is added, DNTP by the DNA polymerase will now start adding the DNTP. Okay, so DNTP will be added. But still, let me just draw a correct diagram. So this is the primer, this is the DNTP, this is the primer, this is the DNTP, primer, DNTP, like that, in this fashion. But still you could see the gap between the primer. This is primer and this is DNTP. Primer, DNTP. primer DNTP. Okay, so like that. So gap is seen. This gap is now filled by a special enzyme called as DNA ligase. So ligase will come and join it. Ligase. Again, the synthesis of the new strand of the DNA is from 3' to 5', correct? No, it is 5. This is 3' and this is 5'. Sorry. Okay, so this is the thing. So 3', just a minute. This is 3' and this is 5', correct? Okay, so again, correct, yeah, correct. Now the replication will be from 5' to 3'. Okay, I was a bit confused actually because of my handwriting. Correct. So correct, so this is actually the polarity, correct? So the new DNA strand will be in the polarity 5' to 3'. Done, very easy, correct? So 5' to 3'. But in this case, the process of polymerization is continuous. So this strand is called as continuous strand, or it is also called as leading strand. Please mention two names. In this case, the polarity, that is 5' to 3', the strand synthesis will be continuous and it is called as leading strand. But in this case, due to many primers are added, that is from 3' to 5' end, so this process is discontinuous, correct? As you know it, why it is discontinuous, and it is called as the lagging strand. What do you call it? Lagging strand. And it was first observed by a scientist, that is the lagging strand was observed by a scientist called as Wakazaki. Hence this strand can also be called as Wakazaki fragments. Wakazaki fragments. These Wakazaki fragments, as I told you, that are joined by a type of enzyme called as ligase. So this is all about DNA replication. Nothing else, actually very simple. If you have any confusion, please let me know about it. And moreover, the mobile phone is old, and because of that the camera quality is also not good. Hope you can see, and if you have any doubts you can contact me. Okay, I'm available to explain the concept again, no issues. Okay, chalo, take care, bye.